We do a lot of crystallography. And the first step in crystallography is to grow crystals of your protein. This is often actually the hardest step of the whole, solving a structure of a protein. So in our lab, we are working with the Maynard lab at UT Austin to engineer these antibody fragments that we can use as crystallization chaperones. Membrane proteins specifically are really difficult to crystallize because there's lots of hydrophobic areas in membrane proteins, and crystal contacts are polar. So you need more polar residues that can form these contacts so that you can form a crystal. What we are trying to do is have these antibody fragments that bind to a specific epitope, and our epitope is EYMPME. It's a hexapeptide. So our antibody fragments recognize this epitope, and then we can engineer that epitope into any protein that we want. And once we do that, then our antibody fragment can bind to our protein of interest, and it should help that protein crystallize easier because antibody fragments are hypercrystallizable, and they add a bunch of polar contacts to the membrane protein that you're interested in. Our first target membrane protein is signal peptide peptidase. So once that structure is solved, that's going to be huge news. It's going to be, really interesting structure to have. But further than that, we hope to use these antibody fragments to solve other membrane protein structures and for other people to use these antibody fragments as well. We hope that we can show the crystallography community that our antibody fragments really do help crystallize these membrane proteins easier, and with our antibody fragments you can solve membrane protein structures faster and more of them. We're hoping that we can just use these to solve many membrane protein structures, and SPP is just going to be the first of many.